Hello, and thank you for joining us. I'm Laurie Cox from Brigham and Women's Hospital and Harvard Medical School, and I'm the moderator of the full scientific session on the microbiome and gut-brain interactions. This presentation is one of 3 pre-recorded videos highlighting work on the microbiome and neurologic disease before all the panelists come together in February during the live discussion. In this video, Rosa Krajmalnik-Brown will be discussing microbiota transfer therapy for autism and lessons she learned from multi-omic approaches. Welcome, Rosie. We look forward to hearing your work.
I am Rosa Krajmalnik-Brown, and it is my great pleasure to be here to present some of our findings from our microbiota transfer therapy for autism, which is a treatment that we did to modify the microbiome in kids with autism and hopefully relieve some of their gastrointestinal and behavioral symptoms. I want to start by thanking my collaborators, Dr. Jim Adams and Dr. Daewoo Kang. They were instrumental for this research, and without them, nothing would have happened the way it did. We have a long list of other sponsors and collaborators too. Autism diagnosis in the United States has been increasing dramatically in the last 20 years. These are data collected from the Center for Disease Control, CDC. The diagnosis is usually based on behavior, and there's no biochemical that can be used for diagnosis. Additionally, there's also no cure or treatment available for autism, and the estimated costs are huge for each individual family and for the state and the whole country. We have been looking at the microbiome as a component of autism, and some of the things that we observe are the lack of diversity in kids with autism. They're probably missing beneficial microbes. So we designed a treatment where we added microbiota from healthy donors. What we did was 2 weeks where we had vancomycin to get rid of the microbiota that was initially in these children. And then there was high dose, initial or high dose treatment. And these children also took lower dose orally for a 7 weeks maintenance dose. We collected fecal samples and we collected plasma samples, and we also collected behavioral and gastrointestinal assessments. The first piece of exciting finding was that we were able to change the microbiome. And here is just one of the metrics that shows that we did that. Here you can see faith PD, which is a measurement of diversity on the y-axis. The highest this number, the higher the diversity it is. On green are typically developing controls. And these are 2 different types of donors. And you can see that the median of the diversity in our children before treatment was much lower than in our typically developing kids. And as the treatment progressed, this diversity increased even more at our follow-ups, which was after the treatment, week 18 and 2 years later. We also look at some specific microbes, Bifidobacterium, which is a probiotic, Prevotella, which is a microorganism that we've been following because it was severely depleted in the first 2 cohorts that we looked at. And the sulfovibrio because we've been looking at sulfate reducers and their activity in the gut. And you can see again here a log of a relative abundance, and on green are typically developing controls. So you can see how there was an increase and the numbers were higher than the neurotypical for all of these microbes at the end of treatment. When we look at GI assessments, gastrointestinal symptoms were rated with this GSRS scale. At the top of the scale on the y-axis, the higher the scale, the worse the GI symptoms are. The number 1 would be a normal gut. So you can see here that the children started with very severe gastrointestinal symptoms, and that after treatment, which is week 10, many of those symptoms were remitted, and some of them were lost at the follow-ups, but they were still much better than in the beginning. The main symptoms that we were able to reduce were diarrhea, constipation, and abdominal pain. The behavior of these children also improved, and we did a few different metrics, but here I'm showing CARS, which is a child autism rating scale. And the reason I'm showing this, it's because this was done by an external evaluator. Our treatment was open. There was no blinded side, so it was an open label treatment. Everyone knew that they were being treated. So there is a possibility of some placebo effect. So it helps that these assessments were not provided by the parents. And you can see here that in the beginning, the majority of the children were in the severe category. And after our follow-ups, only 3 or maybe 4 remain in the severe category, and most of the children improve. Not all of them, but most of the children improved dramatically. For us, it's very important to, based on these results, which were super exciting, even though, like I said, it's an open label study, FDA provided fast track for MTT to be used for autism. My research team is interested in the possible mechanisms that could lead to this interaction between the gut and the brain. And one possible mechanism is the production of metabolites through these microorganisms. And these metabolites would have to travel through the blood, which is one of the reasons why we look at blood for these metabolites. Here I'm showing a PC plot where what you can see is how each dot in this plot represents the whole metabolome of one sample as a whole, as a compact whole. We're not looking at a specific metabolite, but just the metabolome as a whole. And you can see in red circles, the kids with autism before the treatment. You can see most of them here, only a few here, but most of them here. And you can see with blue after 3 weeks, which this would just show the effect of vancomycin because the blood sample was taken before the inoculation with the high dose. And after 10 weeks, which is the green dots. And you can see here that after 10 weeks, these samples group more closely with the squares, which are typically developing controls. So it is exciting first to see that we change metabolites in a systemic way, that the changes in the gut travel through the blood, and we can detect that with metabolites in the blood. And the second exciting finding is that these samples now are more similar to the typically developing children. We also look at specific metabolites, and there were some metabolites that were lower when we compared typically developing kids with autism before treatment, and they changed and increased after treatment, and you can see those with the blue arrows. And we had some metabolites that were higher in the beginning and they decreased after treatment. You can see those in this schematic with the red arrows. On the top, you can see plasma metabolites. We saw more dramatic changes and more statistically significant changes in the plasma metabolites. You can see that we saw this in many different categories, including amino acid metabolism, which is really important for production of neuroactive molecules, cofactors and vitamins, lipids, metabolism, xenobiotics, carbonate metabolism, and purine metabolism. And in the fecal metabolites, significance was more difficult to achieve. Maybe part of this because this is a more heterogeneous matrix. But we were able to see changes in paracresol sulfate, which is a metabolite that we've been following because it's a metabolite from tyrosine microbial metabolism. And it's very similar to 4-EPS, which is a metabolite that Dr. Sarkis Masmanian has been looked into both in mice and in humans. And so with this, I hope I convince you that we did a treatment that changed the microbiome, changed the metabolome in a global way, in a systemic way, and also changed metabolites in the gut. We still don't know what is the mechanism for the changes in behavior and in GI, and we're looking at that, and we're also currently running a blinded study with a placebo arm. With this, I will conclude my presentation, and I will be happy to take any questions.
Thank you, Rosie, for your wonderful presentation. Now, Dr. Emmerin Mayer will join the conversation to further delve into the material. Welcome, Emmerin.
Very nice presentation. Thank you for providing this exciting information about your studies with patients with autism spectrum disorders. So I have a few simple questions that came up listening to your talk. So one is, could differences in individual diets explain the gut microbial differences that have been observed and that you've observed in other studies? So particularly one feature that decreased relative abundance of Prevotella, which is also seen in industrialized societies. And so could you comment on that? Could that, what role could that play in the whole, microbiome, gut, brain abnormality that we see in autism spectrum?
So first of all, that's a great question. Prevotella has been associated with the consumption of fiber. And fiber is one of the things that we in the Western world don't consume enough. And so when you compare the population from the Western world to, for example, Africa, Africans have a lot higher Prevotella than Americans. And part of this could be because of what we eat, the consumption of fiber. So Prevotella is definitely, generally talking, correlated to diet. In our study, we didn't see a strict correlation with diet. And we monitored diet in the beginning, and unfortunately, we didn't have dietary journals after the treatment, which would have been great. That's something that we're doing now, but that would have been great because even though generally the patients said that they didn't change their treatments or the supplements or generally their habits, I would have hypothesized that these children were eating a more diverse diet once they were feeling better.
Yeah, and this is something, I mean, I can only tell from the patients that I see, the autism spectrum patients I see that come to me with GI problems.
Mm-hmm.
And one consistent theme that I always hear from the parents, these heavily restricted diets, that there's so
Mm-hmm.
many intolerances, even either based on the texture of food or certain things, that autistic. So they create a very special diet for themselves.
Yeah, and so the more restricted the diet, the microbiome will be more restrictive too. So we see here the increase in diversity probably at the end, and even after 2 years, which we saw this increase that was even higher than the donors and the typically developing children, must have been also because they were consuming healthier food or a more diverse menu.
Yeah, and this is obviously a very important point. I mean, so I was going to comment on in a positive way. So your studies have not only shown one of the very few that for a chronic medical condition, psychiatric condition, have shown that you can overcome colonization resistance of the microbiome, which is amazing considering the efforts that have gone in IBD and in obesity, and they all have basically failed to show that. So, and the other thing is, that it's remarkable that you had this 2-year, this persistent benefit. So one thing I do, as we just talked, how much of that would have been that kids overcome this extremely restrictive diet, and then their GI symptoms may improve because they no longer they treat both them gut microbes better and have a better gut health. And so yeah, this was basically just a comment. And I mean, do you have any thoughts about this, why you have been able to overcome the colonization resistance and others have not?
So one of the things that when I have more time to speak, I usually mention is that my belief as an environmental engineer and as a person that works with microbes in their environments is that the key is to change the environment. Because if you have an environment that it's not good for beneficial microbes, then it doesn't matter what you do. Whatever you put in there, the good microbes are not going to thrive if the environment is not right for them. So somehow with the treatment, we achieve a change in environment, which led to these children being able to acquire and grow beneficial microbes. So the treatment was very intense. It wasn't just the FMT. We had these 2 weeks of vancomycin, and then we emptied the gut. And then we also, these children took proton pump inhibitors, which I can't think that they're good. And you probably will tell me that they're not good for the gut, but somehow these together with the new microbiota achieve a change in environment. And I think that's the key. You have to change the environment for good microbes to thrive.
Which is what we know from other ecosystems.
Right.
You can't introduce new species unless you get rid of the compromised ecological environment. Right. Yeah. So another question, so you already answered this. So you will in the future follow the dietary changes. Did you follow the behavior of the parents during this? I mean, this must be obviously a remarkable positive experience for the parents, and I could imagine with decreased anxiety and more optimism, less depression. Did you look at that, and did that have any relationship with any of your
That's a great question. Also, we didn't follow them. I got comments from our study coordinator, and I have seen a couple of the parents interviewed by the news, because sometimes when the news clips want to interview the children or the parents are very excited to talk and to share their experiences. And someone who criticized our study said that maybe what happened was that the parents were more relaxed and better and more hopeful, and that these children were getting so much attention that that's why they thrived. And you know what? If that's what it was, that's what every parent needs to do, and that's all I have to say. But we think there was more than that, and we proved that there was a change in the microbiome, which is one of the reasons why I think those microbiome and metabolite measurements are so important. Because if we had done just the behavior, we wouldn't have the microbes and the biochemistry to show that there were systemic changes happening.
Yeah.
Yeah,
So
I
mean, for me personally, I'm a big believer in the placebo and nocebo response. So, and I link it to, the brain-gut communication.
The one that goes from the brain to the gut, and not
yeah.
the other
So
way around.
if somebody feels better, both the kids and the parents, it will have an effect on the gut as well, and the microbes as well. So I think it may just be one component.
So I'm not denying that could be a component, and I agree with you, but our results are still pretty exciting.
So one question I have is related to Sarkis's presentation about the 4-EPs, the metabolite that people have done a lot of studies on, and that are elevated in about, I think, a third of patients with autism spectrum. And the close chemical relative is P-Cresol. And so in one of your studies, you saw an increased level of P-Cresol in the stool, but not in the blood. So do you think this could mean you were looking at some of the different population that did not have these elevated 4-EPs levels, or I mean, how do you put this together? So obviously his presentation is very strongly suggestive that in a subset of patients this plays a role, as it does in mouse models. But if I understood your presentation correctly, you didn't really see that.
No, we did not. We saw paracresol sulfate, which is a relative of 4-EPS. If you see the pathway, it's also a product of the tyrosine metabolic pathway. And we saw tyramino sulfate in a blood that was statistically significantly different, and it's also part of that pathway. So we did not see 4-EPS, and we looked for it because I have known Sarkis for a while, and I have known that he's been looking for it. So we saw it, but we didn't see a statistical significant difference. Keep in mind that our study was 18 children, so it might be that if you look at his data, it's only like a certain percentage of the population that is higher. So it's almost bimodal if you look at his data. So it could be that we didn't have the right population to see it, that maybe with a larger sample size we would see it. But we did see products of that tyrosine pathway, which I think is very interesting.
Okay. I mean, you have already, shown more progress in this field than anybody else in terms of, like potential therapeutic implications. So where are you going to go from here with this? I mean, obviously, your studies haven't been controlled up to now, so I imagine you would go in the direction of
So we're running right now an adult study funded by DoD that has a placebo arm. We're continuing to look at which microbes are changing and which metabolites are changing, because ideally we would like to understand how is this working. And I personally would like to be able to craft a cocktail of microbes, because I don't think it's one microbe. I think it'll be a community that you can grow in the lab, because that would be easier, than relying on fecal samples. It would be easier to produce, and to have a good supply, because we have right now people lining up to participate in our studies. Like we had more than 1,500 applicants for our previous study, and we can take 50, And I don't know how you would take that many, and how would you supply, you have to produce it and you have to screen it, and it would be easier if we could just produce it in bioreactors. But for that, we would need to understand which are the important microbes that make a difference, or even better, if we find some metabolites that are missing that we could just supply as a supplement.
Yeah.
You know.
I mean, our group, we have also funding. So I'm a co-PI on a study with Lisa Azadeh at USC on a DoD-funded study on brain features. It's a cross-sectional study. And as you know, there's clearly a lot of findings, both in baseline resting state, but also in evoked responses to various stresses and tasks. And so it's something, and we've talked about this in the past. I mean, I would love to see that. And are there brain changes in those individuals that have gotten better with this intervention? With the
you and I spoke about this, scanning these children after the intervention, before and after. You and I spoke about it, but I think the logistics would be. You and I wrote a proposal together, but the logistics of actually implementing, especially when we're not in the same state. But I think we should do it.
Yeah, we should do it now that you have these exciting preliminary data. I think it's it'll be a lot more realistic to do it. So, do you think? The last question. How far away? I mean, this has been such a devastating disease, and it keeps increasing. Or has been increasing dramatically. How far away do you think, based on your findings and Sarkis's amazing findings in his animal models, how far away do you think are we from developing realistic, practical treatments for ASD?
I think that with FMT we're pretty close, though, because we're doing right now a phase 2. Then we would need a phase 3, assuming that the results hold. Mm-hmm. It's not that far. And like I mentioned on my presentation, FDA gave us fast track. So that means that they will look at the data quicker and take off a little bit of the bureaucracy, hopefully. Mm-hmm. But I'm saying really, really soon. That's still, as you might know, it's 5 to 10 years. It's not, it's not like the COVID vaccine that was within a year, right?
Yeah. Have you noticed from feedback from parents? So I imagine, now that this is in the public domain, that a lot of parents take it in their own hands and do fecal microbial transplants. Have you ever heard from parents that did it on their own, that they got similar kind of benefits as you have in your study?
I haven't heard, but anytime that reporter interviews me, when they said, "Did I miss something?" I always recommend to the parents that they don't do it that way, because the material needs to be well screened. And if you don't do that, you can end up with a bad GI infection. Mm-hmm. So ideally, I don't recommend that people do it by themselves and without medical supervision. And so I haven't heard much, because that's not a practice I recommend.
Yeah. Okay.
But I'm sure they do it. I know there's a clinic in Mexico that charges a lot of money and does it. Maybe we should plug that.
The key issue is this whole thing that we talked about in the beginning. An essential part is ecosystem restoration. Portion. It's not just the transplantation. Right. It's
I agree.
So.
Yeah. And so it would be great also if we could achieve that ecosystem restoration just with diet. I'm a huge fan that if we can figure out how to do that just with diet, that would be even better if we could do that. We haven't figured out how to, but if we could, I think that would be great.
Yeah, and there's one question. Also ask Sarkis about this. So this disease doesn't start, in a 1-year-old infant. It's already probably the first beginnings of this, and abnormal brain development in utero. So dietary interventions in pregnant mothers, preventing this whole toxemic, this metabolic endotoxemia, and decreasing another risk factor in women that have already had one autistic child. It's interesting that has not really been done. So, I was wondering, if that's something, I mean, have you heard about this in your interactions with families, that people have talked about doing studies like that?
I haven't, but there's, if I remember correctly, even some papers about how high-fat diets or obese moms actually have a higher risk of having
they
kids
have a
with
higher,
autism. Yeah.
yeah. I just haven't seen the intervention. You know, if you,
Yeah.
I
So
mean,
do it the other way. But when we're talking about moms, there's also try to minimize C-sections because that's another intervention that sometimes is needed, but when it's not needed, it's reducing the microbial diversity that it's needed, and try to promote breastfeeding, which, you know, we do as much as we can. Yeah.
Okay, Rosie, I think you've answered, you know, I mean, we could go on in this conversation for a long time.
I know. It's fun, right?
Fascinating area. And
It's been fun.
thank you very much for your, you know, very interesting talk and the conversation we had.
Thank you again, Rosie, for your remarks, and Emmerin for joining in the discussion about the material. Just a reminder, this is but one of 3 spotlight videos focusing on the individual work of the speakers before they all come together in February in the live panel, Microbiome and Gut-Brain Interactions. Please check the AAAS annual meeting website for the panel's date and time. Again, thank you, Rosie, Emmerin, and thank you, the audience, and we'll see you live in February.